Welcome to our first NBEO Go bonus review video. This video is meant to get you in the mindset to study molecular biology, biochemistry, and genetics. This all ties into systemic health, which we know is a large part of the Part 1 test. Before we begin, it's important to understand the central dogma of molecular biology. And why is it so important to understand the central dogma? Well, one reason is it is specifically listed on the NBEO posted outline for Part 1. In fact, all of these videos are based off of the outline that's supplied by the board. Anyways, the central dogma describes the flow of genetic information within a normal biological system, where DNA is transcribed into RNA, which is then translated into protein. This sequence is fundamental to how genetic information is expressed. So to understand how proteins are made, we have to start with DNA, which of course is the blueprint of life, and then go on to other steps. These two main steps are transcription and translation. We'll break down these steps in detail, but first, it's very important to note that transcription starts in the nucleus of the cell. I think it's pretty important to know that transcription starts in the nucleus of the cell because translation occurs in the cytoplasm. And it'd be a pretty easy board question to get right if you knew where each of those steps takes place. Well, the first step of transcription is called initiation. During initiation, mRNA polymerase, thank you, graphics team, mRNA polymerase is the enzyme that will do the actual mRNA creation. It binds to a region called the promoter region on a gene of DNA. I think of the promoter region as almost like a runway that a plane would land on, only when it comes to DNA, it's where the RNA polymerase will land on so that it knows that it's creating the correct gene. So some common examples of promoter regions include the ta-ta box, which is rich in the nucleotides thiamine and adenine, and there's also the CAAT box, which I call the cat box, and the GC box. So all of those are just examples of promoter regions that the board's questions might be about. There are such things also called transcription factors. And I think of transcription factors as almost like on and off switches that control when DNA is or is not expressed. So there are a lot of things that are involved in the actual initiation phase of transcription. So lastly, there are these regions called enhancer regions on DNA, and they do exactly what their name says. They enhance the transcription of DNA. So that's initiation. After initiation is elongation, and that is when the RNA polymerase reads the DNA template strand and creates a pre-mRNA molecule by adding complementary RNA nucleotides. And we call it a pre-RNA molecule because it will go through a lot of other processing, as we'll see, before it is actually translated into protein. I think it is a good time to kind of take a step back and remind ourselves that there are multiple types of polymerases. In this case, RNA polymerase is what makes the mRNA, but there is such thing as DNA polymerase as well, and DNA polymerase is involved in DNA replication, not DNA transcription. I would hate to be taking Part 1 and forget which polymerase does what, so it's a good thing to remind ourselves of. So during the process of elongation, we have this concept of complementary base pairing. Cytosine always binds with guanine, and guanine always binds with cytosine. Thymine and adenine can be a little tricky. The reason being is DNA has thymine, however, RNA does not have thymine. Instead, it has something called uracil. So whenever we see an adenine on DNA, we know that that adenine will bind with uracil on RNA. Whenever we see a thymine on DNA, we know that thymine will bind with adenine on RNA. So it gets a little tricky. In this slide, you can see kind of the complementary base pairing, thymine binding with adenine, adenine on DNA binding with uracil on RNA, cytosine with guanine, just to kind of remind us of how this situation works. Because again, it would be a very easy boards question to ask for a complementary base pair of either tRNA, mRNA, and DNA. And a really easy way to get that question wrong is to forget that RNA never has thymine. It has uracil instead. After elongation is termination, and that's just when the RNA polymerase reaches a terminator sequence on the DNA, and that just signals that, okay, the gene is done, our mRNA is done, RNA polymerase is done doing its job. Now, a lot of errors can occur during transcription, leading to mutations. So, for example, in a point mutation, there's a single nucleotide change. Let's say, for instance, in this slide, we see that guanine is supposed to be binding with cytosine, but maybe the RNA polymerase accidentally put adenine there instead. And all of the base pairs are correct with the exception of that adenine. So this is the original sequence, and this is the sequence that's supposed to be made, and you see that there was just a mistake here by the RNA polymerase that put the wrong nucleotide, and so only this one section is affected. This front section of these other 3 units, which we call codons, 3 units at a time, are just fine. Nothing, no harm, no foul on those. There are other mutations that can cause what's called a frameshift mutation. And that happens when instead of simply putting in the wrong base pair, the RNA polymerase may accidentally add in an additional base pair. So here we see it's supposed to be AUG and then C, and we see that it says AUCGC. It accidentally added an extra cytosine that should not have been there. And because of that, there's a complete frame shift where the 3 corresponding codons are never correct. The same thing happens with a deletion. So I would say additions and deletions are some of the worst mutations as opposed to transcription errors where just maybe one nucleotide was put into the wrong space and replaced the other, have less of a problem down the line when it comes to protein creation. So after elongation and termination, when transcription is over, it's time for the mRNA strand to be further changed. So one of the ways this happens is coding regions, which we call exons, are put together. So what I mean by this is when we look in this picture, we see the RNA strand that is created from that complementary DNA. We can see those complementary nucleotides. And here, an enzyme called a spliceosome is cutting that RNA up into pieces and trying to edit it to what it really wants it to be. So there it has removed a section, and that removed section we actually call an intron. And the way I remember it's called an intron is because introns interrupt what we actually want. So we're going to remove them. So the intron is now gone. And that leaves coding regions called exons left behind. And the way I think of exons is the exons are the expressed regions. So by splicing the mRNA, you can actually create various different types of proteins because they can splice out different sections and rearrange the exons. So after splicing is done, the mRNA usually requires the addition of a 5' cap, which is just a guanine nucleotide, basically, that's added at the beginning of the mRNA. And then there's the addition of a really long sequence called a poly-A tail, which is about 250 adenine nucleotides that are added to the end of the mRNA. It's added by an enzyme called poly-A polymerase. So it kind of says in its name what it does. And it's thought that perhaps this additions are to help the mRNA during its journey from the nucleus to the cytoplasm so it doesn't denature or become damaged. So after initiation and elongation and termination, this mRNA is now ready to travel to the cytosol, to the ribosomes. And at the ribosomes, it's ready for the next step of protein formation, which is called translation. And this occurs again in the cytoplasm at the ribosomes. And ribosomes can either be free-floating or attached to the endoplasmic reticulum, which we call the rough endoplasmic reticulum if there's ribosomes. Otherwise, it's called the smooth endoplasmic reticulum. During the first phase of translation, which is called the initiation phase, there's a small ribosomal subunit that binds to the mRNA. And then a tRNA, which is nothing but a 3-nucleotide anticodon sequence attached to a single amino acid. So then this tRNA binds to its complementary codon. So in the case of protein synthesis in humans, the first nucleotide sequence, or the start codon on the mRNA, is always AUG. The anticodon, of course, for AUG, which is going to be on the tRNA, is UAC. Because remember, RNA never has thymine. It only has uracil. So the tRNA will bring this UAC to the complementary AUG on the small subunit of the ribosome, and that is always attached to the amino acid methionine. Methionine is always the first or the starting amino acid of a growing polypeptide chain. And we call the start of a protein a polypeptide chain, which is nothing but a sequence of a bunch of amino acids bound together by peptide bonds. After the initiation phase, where that initial tRNA binds, we have the elongation phase. And this is when more tRNAs come in and start forming peptide bonds to that initial AUG sequence. Now, this is kind of a part of confusion for me when I was studying for boards. Ribosomes have these 3 sites called the A site, the P site, and the E site. That initial methionine here, it always binds to the P site of the ribosome. Any other tRNA after methionine, rather than binding to the P site, will initially bind to the A site. So it enters the ribosome at the A site and then travels to the P site where it forms a polypeptide bond. So you can think of P as if it stands for a polypeptide. And then after that site, it moves to the E site of the ribosome, which pretty much means the exit site. So remember, methionine, AUG, that is the first sequence of mRNA and the first amino acid on tRNA to bind. It will bind to the P site first. All other tRNAs will go to the A site, and then the P site, and then the E site. The last phase of translation is termination, and this is when a stop codon, which is usually UAA, UAG, or UGA, is reached, and then the ribosome releases the completed polypeptide, which will eventually fold into its functional form. So, interestingly enough, and something that's going to be important to remember is, the ribosomes in bacteria have different subunits than the human ribosomes. So in humans, there's a small ribosomal subunit, which we call 40S, and then a large ribosomal subunit, which we call 60S. And this forms an 80S ribosome. Now, how in the world does 40 plus 60 equal 80? I don't know, but that's just the way it is. Well, in bacteria, the small ribosome subunit is 30S instead of 40S. The large ribosomal subunit is 50S instead of 60S. And the whole ribosome is called a 70S ribosome. Again, how does 30S plus 50S equal 70S? I don't know. But it sounds like a really easy board question to get wrong because you forgot that for some reason in the microbiology world, 30 plus 50 equals 70 instead of 80. So these differences are actually quite important because they are one of the ways that things like antibiotics can specifically target bacteria and their protein translation and inhibit bacterial protein synthesis without affecting eukaryotic cells. So it's funny how you'll see that boards in part one can tie molecular biology with biochemistry and genetics with other topics like microbiology and pharmacology. So a good foundation will help you gain confidence that you need to pass part one. So now let's go ahead and end with exploring the different levels of protein structure. So a protein's primary structure is very easy. It's just the sequence of amino acids and a polypeptide chain. And this sequence is pretty much determined by mRNA and ultimately the DNA sequence that it came from. So again, the central dogma. The secondary structure of a protein includes alpha helixes and beta pleated sheets. These are stabilized by hydrogen bonds. An alpha helix is coiled in structure and has hydrogen bonds in between the backbone atoms. And then a beta pleated sheet is folded and sheet-like, and it also has hydrogen bonds between segments of the polypeptide chain. The next structure of a protein is a tertiary structure, and it's more of a 3D shape, but it's important to note that in tertiary structure, this 3D shape is a single polypeptide chain. And there's lots of different interactions, hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interactions. So a lot of biochemistry involved in the folding into a tertiary structure. The tertiary structure does, give these proteins their functionality. And you'll find that if you look at a tertiary structure of a protein, many times the hydrophobic amino acids hide in the center of the newly folded protein, and the hydrophilic amino acids are around the exterior of the newly folded proteins. Also, you'll notice that proteins don't necessarily fold on their own. There are special proteins that can help called chaperonins, which assist in the folding of these amino acid chains into their final structure. Now, some proteins have a quaternary structure. So remember I said that tertiary structure is just the folding of that amino acid chain into a complex structure, but it's only one polypeptide chain. In quaternary structure, it's the same thing, only there's multiple polypeptide chains or subunits that form the functional protein complex. All together, again, folding takes place spontaneously because of all these interactions, but the chaperonins do help with protein folding. Proteins can be categorized based off of their solubility and function. And this is definitely something you want to remember. Boards actually puts on their review outline that we are to know some of the basic microbiology-based protein structures. So let's go over some examples. So, fibrous proteins, those are typically structural in nature. They don't really have much of a function, and they are typically water insoluble. So they tend to be hydrophobic. Examples are collagen, which provides strength and elasticity to connective tissue. Collagen is a triple helix. There are 3 polypeptide chains wound around each other. So that's structure, that triple helix, you should know that. Elastin is another one of these fibrous insoluble structural proteins, and it allows tissues to stretch and, most importantly, return to their original shape after stretching. So elastin is more or less composed of random coils instead of 3 spirals, and is just, as its name says, highly elastic. Keratin is another structural, insoluble, structural protein. It's found in hair, nails, skin. It provides protection and strength. Keratin is kind of tricky because it can be alpha keratin, which has coils, or it can be beta keratin, which is that beta pleated sheet formation. Keratin is often held together by disulfide bonds. The next type of structure outside of these fibrous, insoluble structural proteins are the globular, soluble, functional proteins. So you see the contrast. Fibrous proteins are generally insoluble and structural in nature. Functional proteins, globular proteins, are usually soluble and have some type of function to their nature. An example of this is hemoglobin, which we know carries oxygen in the blood and transports it from the lungs to the tissues. Enzymes catalyze basic reactions. Those are also functional, soluble proteins, globular proteins. And enzymes, as you know, basically increase a reaction rate without being consumed. Examples include amylase, that's an enzyme that digests starch, or lactase, the enzyme that digests lactose. Antibodies are part of the immune system. Those are also functional soluble proteins, and they identify natural pathogens such as bacteria or viruses. And each antibody actually has a unique binding site that matches a specific antigen. So these proteins can become very specific. So connecting protein function to real-world scenarios in optometry can enhance our understanding. For example, if we understand the role of collagen in the eye sclera or the cornea, it can help diagnose connective tissue disorders affecting the vision. Anyways, that's all for this basic review video on protein synthesis. These concepts are carried throughout the NBEO outline. Stay tuned for more videos to help you ace your Part 1 boards examination. We hope that these help you. Good luck.